Hello there everyone. My name is Avishek Kataria. Our study, the MLL WDR5 complex, promotes midbody recruitment of KIF2C for microtubule depolymerisation and furrow initiation during cytokinesis is the topic of discussion today. MLL or KMT2A, facilitated by partnering proteins such as WDR5, carries out its well-known function of histone modification-based gene regulation. Our findings, however, have demonstrated that MLL plays a non-canonical role in cytokinesis, the last phase of cell division. We begin by examining the location of MLL and WDR5 proteins. MLL was visible to us as a dark zone protein and a stem body protein. Through SIM microscopy, we were able to visualize it as a single ring. WDR5 was also visible to us as a flanking zone protein and a dark zone protein. We observed that MLL WDR5 deficient cells struggled to form a normal midbody, leading to binucleated cells, which is an indication of faulty cytokinesis. Additionally, seen were chromosome bridges and an enlarged intercellular bridge. However, MLL-WDR5 does not act in isolation. We have previously identified the MLL-WDR5 KIF2A connection on the spindle pole in our lab. We found that WDR5 was interacting with sibling members of KIF2A, KIF2B, and KIF2C, which are known to localize to midbody. Pull-down and reciprocal pull-down experiments utilizing GST-bound proteins verified this relationship. Additionally, we found that KIF2C could interact with MLL regions that contained the FYRC and Z domains. Similar results were also obtained using HeLa cell lines that expressed lab type KIF2B and KIF2C. As cells enter telophase, KIF2C accumulates at the midbody where its depolymerase activity helps to drive the final steps of cell division. When MLL or WDR5 is knocked down, KIF2C localization is reduced at the midbody. A decrease in KIF2C at the midbody causes problems in midbody formation, leading to increased intercellular bridge width and binucleated cells. To better understand this phenotype, we utilized live cell imaging, and it revealed to us that in healthy cells, furrow initiation happens swiftly and which quickly transitions to midbody formation. However, in cells lacking MLL or KIF2C, this process is delayed, disrupting normal cell division. Progression to midbody formation from furrow initiation is occurring at a slower pace. Interestingly, the depolymerase activity of KIF2C is essential for proper midbody formation. Cells expressing a mutant KIF2C which lacks this activity shows abnormal midbody structures and delayed furrow initiation. Because dynamic microtubules are needed for correct central spindle positioning and cleavage furrow formation, the team examined the localization of RAGAP1 and aniline. Control cells showed compact RAGAP1 at the ICB, but MLL or KIF2C depleted cells showed misaligned or dispersed RAGAP1. Similarly, aniline failed to progress to a constricted contractile ring in depleted cells, remaining at an early furrow stage. Overall, the results demonstrate that MLL-WDR5 dependent recruitment of KIF2C is essential for microtubule dynamics that enable proper mid-zone compaction, spindle organization, and completion of cytokinesis. Thanks for watching and read our article for more details.